All right, so this video is going to introduce the complement system, which is one of the innate immune mechanisms that human bodies have. One of the first line of defense to recognize and attack pathogens when they're present in the body. So the complement system, it can be a little complicated. There are lots of proteins involved, a lot of protein-protein interactions, a lot of different mechanisms to activate complement, but this series of videos is going to walk you through the entire complement system, which again are very robust mechanisms to identify pathogens and to remove pathogens from the body. So let's talk about what the complement system is. It is a series of proteins that are made by the body, about 30 or so proteins. These are soluble proteins, or we're going to find them in our fluids. Now, remember, our extracellular fluids, our interstitial fluids, are the liquid portion of blood, the plasma, the lymph. So, if there are pathogens in our fluids, these extracellular fluids, these pathogens will be interacting with complement proteins, and the complement proteins will help recognize and remove the pathogens. So complement proteins, some examples of names of complement proteins are proteins like C1, C2, C3, C4. You can see the list there. Also, some proteins whose names are factor B, factor D, factor H, factor I. These are just some of the complement proteins that we're going to encounter as we learn about the complement system. Where do these proteins come from? Well, they're made by cells in the liver. Interesting part of the immune system, the liver, but the liver makes complement proteins. And the thing about these proteins are that they are constitutively expressed. So, what does that mean? Remember, the innate immune system, right? The innate immune mechanisms are, you're born with them, they're present, and they're ready to recognize and attack pathogens, you know, very quickly. So if you want something to act quickly, you need to have it ready to go. So complement proteins are present in the body at all times. So you don't need an infection to have these proteins made. They're circulating in your fluids, ready to recognize a pathogen. So that's what we refer to as constitutively expressed. All right. So now let's talk about, what do these proteins do? Good question. So here I've got a pathogen, I got a virus, I got a bacteria. That's how I draw them. So if we're going to cut to the chase, we're going to talk about the fact that complement proteins, not all of them, but some of them, can be covalently attached to pathogens, to the molecules on the surface of the pathogens. Complement proteins can react with proteins or sugars or lipids on the surface of pathogens and mark them for destruction. So we call this process complement fixation, complement proteins being fixed to a pathogen surface. So we want to introduce that concept. How that's going to happen, we'll get into the nitty gritty in later videos, but we want to introduce that concept. Now, how did the complement proteins activate to get attached? So there are multiple mechanisms to activate complement. In fact, there are three pathways we talk about which complement could be activated. There is the alternative pathway of complement activation, the lectin pathway of complement activation, and the classical pathway of complement activation. All three pathways can lead to complement fixation, but they're all going to activate a little differently. They're going to involve different proteins, different triggers that are going to be involved in recognizing the pathogen. If you remember about the themes of immunology, one theme is the immune system has to recognize this thing doesn't belong in the body. So these three different pathways use different recognition mechanisms to recognize a pathogen. But once they've recognized the pathogen and activated, then complement becomes fixed to the surface of the pathogen, marking it for an immune response. And what kind of immune response are we talking about here? So we're going to talk about three different immune responses that are going to help remove this pathogen from the body. One is when complement is fixed to the surface of pathogen, it helps improve the process of phagocytosis. If you recall, phagocytosis involves cells like phagocytes, macrophages, neutrophils, taking a pathogen it's in its vesicular system and destroying it inside compartments inside the cell. So when complement covers a pathogen, when complement's fixed on a pathogen, the process of phagocytosis is much more efficient. And we have a term for this called opsonization. When a pathogen is covered with something that makes it even more attractive to um, phagocytes, that is called opsonization. And we'll see later how antibodies can play a role in opsonization, but complement can play a role in opsonization. Okay. Secondly, complement, when it's fixed to the surface of a pathogen, will trigger the production of something called the MAC, the membrane attack complex. And this can actually drill holes into the membrane of pathogens, basically deflating it, having all the contents leak out. So, membrane attack complex formation will help destroy pathogens. And the third mechanism is inducing inflammation. When complement is activated, inflammation actually can ramp up quite a bit. And remember, the inflammatory response is a very positive response to help fluid flush the pathogen into the lymph nodes, to help bring blood and cells and more proteins to an infected site. So, all three of these effector functions will help clear the pathogen. So, again, going back to the themes of immunology, there's recognition and there's effector function. So the complement system will require us to understand and see both of these. Before we get into the biochemical mechanisms behind complement activation and complement fixation and a complement effector function, we have to review some concepts from biochemistry. Recall from biochemistry, you learned about enzymes. So we're going to reintroduce a type of enzyme called a protease. If you recall, a protease, ASC, it's an enzyme. It's a type of enzyme whose function is to, now recall, all enzymes typically have a substrate, right? If it's an enzyme, enzymes act upon its substrate. So here I've drawn a substrate, and I've drawn a little substrate binding pocket because enzymes have substrate binding pockets. And going back to what a protease is a type of protein whose function is to cleave another protein. So if this protease acts upon its substrate, which is a protein, so we have a protein acting on another protein, then the resulting action upon the substrate is to cleave it. And this can be a very specific cleavage between certain peptide bonds within the amino acid sequence of that protein. And now you have the products of the reaction, which are two chunks of this original substrate protein. So that's just a review of what a protease is. A protease doesn't necessarily cleave every protein. Proteases can cleave very specific proteins, and we're going to see that when we learn about the complement system. So just learning about proteases. They function, right? Proteases are part of the digestive system, breaking down your proteins. That's not what we're talking about here when we're talking about proteases. We're going to see a different function for proteases. They're going to break down specific proteins into specific products. And that's important for us to know when we learn about the actual molecular mechanisms behind complement. So just want to remind you about proteases. The um, the reason we're talking about that in this video is because many complement proteins are proteases. They will function to cleave another protein, and that's going to start a chain reaction. And we'll get to that in the next video. So some complement proteins are proteases. It'll be important to know which ones are which. The other thing I wanted to introduce in this video, a reminder from your biochemistry knowledge, is a term called a zymogen. A zymogen refers to often an enzyme that might be present in an inactive form. So the body might make an enzyme, but it's in an inactive form. So it's not going to cleave. In this example, if a protease is a zymogen. It's not active. But something might trigger a protease's activation, causing it to go and cleave its substrate. So a number of complement proteins, which are proteases, are also zymogens, which means they're going to go from an inactive form to an active form. I just wanted to reintroduce these concepts, remind you of these concepts from biochemistry before we delve into the specific molecular mechanisms behind complement system activation and effector function.